Hello my dear friends, welcome back to CSIR at home. If you are new here, please subscribe and share it, okay? Thereby you can help your friends to get CSIR, okay? In this video, we can study about fungal resistant plants. This picture you can see, this one is a fungus infected plant. The phytopathogenic fungi cause extreme damage and loss of cross productivity. At present, what is the major way of controlling the damage and loss result from the phytopathogenic infection? What is the major way of controlling? Use of chemical agent. But if we use this chemical agent, that may lead to environmental pollutions and also cause serious illness. Here comes the need for transgenic plants. Okay, fungus resistant transgenic plants. Have you studied about pathogenesis related proteins? Pathogenesis related proteins, that is PR proteins. Okay, when plant often responds to fungal invasion, fungal invasion or other environmental stress, how they are reacting by converting special molecules. Okay, this induces the synthesis of pathogenesis related proteins. For example, glucanases, chitinases, then protease inhibitors, etc. These are known as pathogenesis related proteins, PR proteins. Okay, here you can see in this picture, these are fungus, this blue. When the plant infected, what happens? How the plant responds to fungal invasion? It converts the conjugated form of salicylic acid to salicylic acid. Okay, first in plants, the salicylic acid is present as a conjugated forms. It is converted into salicylic acid. And this salicylic acid induces the expression of NPR1 gene. Which gene? NPR1 gene. And this NPR1 gene produces some proteins. And those proteins induces the pathogenesis related proteins. These proteins includes beta glucanases, chitinases, then protease inhibitors and thaumatin-like proteins. Okay, thaumatin-like proteins. These are enzymes and this will help the plant to fight over the fungal resistance. Okay, this is pathogenesis related proteins, PR proteins. And this chitinase, for example, the chitinase. What is chitinase? The chitinase is an hydrolytic enzyme. Okay, this chitinase can hydrolyze the beta-1,4 linkages of N-acetyl D-glucosamine polymer, that is chitin. The chitinase can break down chitin. Chitin is the component of many fungal cell walls. Okay, now understood. If you want to read further, you can refer to CSIRNET, page number 304. Okay, PDF form of book is available in our Telegram channel. Okay. Now we can study how to create the genetic engineered fungus resistant plant. Now we study the mechanism. How we can create the transgenic plants that express high levels of one or more PR proteins. Okay, the expression of PR proteins can be increased. For example, chitinase, which can hydrolyze, we studied, that is beta-1,4 linkages of N-acetyl glucosamine. Okay, that is the component of many fungal walls. The first is high level production of this enzyme, that is chitinase. Then overexpression of NPR1 gene. Okay, the NPR1 gene that we studied, this NPR1 gene induces which one? Pathogenesis related protein. So we can also overexpression, that is overproduction of NPR1 gene. This is also lead to generation of broad spectrum disease resistance against both fungal and bacterial pathogens. Then another approach is transgenic plants with increased production of salicylic acid. Okay, we have studied initially the salicylic acid is present in conjugated form. When a fungus infects a plant, it produces salicylic acid. So we can overproducing this salicylic acid. Okay, overproducing salicylic acid. How the salicylic acid is produced from chorismate. Chorismate is an intermediate in tryptophan biosynthesis. Chorismate. Now we can check the biosynthetic pathway. This pathway, how salicylic acid first is produced from chorismate. Chorismate is converted into isochorismate. Okay, isochorismate with the help of which enzyme? Isochorismate synthase. Then this isochorismate is converted into salicylate, that is salicylic acid with the help of isochorismate pyruvate lyase. Isochorismate pyruvate lyase. This is the pathway. So how we can produce salicylic acid? How we can increase the production of salicylic acid? We can take these genes from bacteria, that is two bacterial genes for salicylic acid biosynthesis. And the salicylic acid biosynthesis gene, which are the isochorismate synthase and isochorismate pyruvate lyase. These two enzymes can be fused with chloroplast targeting sequences. Chloroplast targeting sequences. Why? Because this chorismate is present inside the chloroplast. Okay. So we take these bacterial genes for synthesis and then fused with chloroplast targeting sequences from gene for small subunit of ribulose bisphosphate carboxylase. Okay, we are taking this chloroplast targeting sequences from ribulose bisphosphate carboxylase. So the plants always produce PR proteins because salicylic acid is always available inside the plant. Okay, understood. Next is with chitinase. Okay, always expressing chitinase. Okay, then the transgenic plant that have been engineered to constitutively express chitinase under the control of cauliflower mosaic virus 35S promoter. Okay, and the examples of these transgenic plants including rice, tobacco and canola. Then the other strategy is the genes for two different antimicrobial peptides. The antimicrobial peptides, those are also effective against fungi. Okay, antimicrobial peptides. The gene for two different antimicrobial peptides. Why we are taking two different antimicrobial peptides? For enhancing the activity against pathogens. So two different antimicrobial peptides, one from radish and another from mold, that is Aspergillus giganteus. And these two antimicrobial peptides are fused with chitinase. Chitinase is taken from wheat. The fusion is first two antimicrobial peptides, then it is fused with chitinase to a single chain Fv antibody. And this can bind to Fusarium cell wall. Fusarium, it's a fungus, Fusarium cell wall. In this picture you can see this is antimicrobial peptide number 1, microbial peptide 1, then microbial peptide 2, then chitinase. Okay, these three are fused form. And this is antibody. Okay, you can see, and this microbial peptide is bind. These are cell wall, fungal cell wall proteins. This can bind to cell wall and degrade it. Okay, chitinase is there. Then tobacco osmotin. Tobacco osmotin has been expressed in transgenic potato, providing resistance against Phytophthora infestans. Okay. Then tomato plant expressing grapevine gene for stilbene synthase. It gives protection against botrytis cinerea. Then hypersensitive response. What it is? It is a physiological defense mechanism Okay, similar to pathogenesis related protein. The hypersensitive response. It is a physiological defense mechanism. It is used by plants to repel attacking, used by plants against pathogens. Here you can see this is pathogen. It produces an AVR gene. That is AVR genes. The pathogen produces AVR genes, and this AVR gene is come and bind to the receptor. The AVR gene bind to the receptor, okay, and it lead to the activation of hypersensitive response. It includes phytoalexin biosynthesis, lignin biosynthesis, salicylic acid biosynthesis, and biosynthesis of hydrolytic enzymes. That collectively we can call it as systemic acute resistance. Okay, then how the transgenic plants can be created? That means if this AVR gene is present inside the plant cell, AVR gene is a part of fungus. If it is present inside the plant, that means it will produce these responses. Okay, so the transgenic plants have been created. For example, tomato plant have been transformed with AVR9 gene from Clazosporium fulva. And these tomato plants showed great resistance to wide range of fungal, bacterial and viral diseases. If you like this video, please share and subscribe. Don't forget to share to your friends. Okay, thank you.